Sorry, I'm
So
a
we,
little late.
yeah, no worries at all. So we have been talking about evolution by a mechanism of natural selection. We've only talked about natural selection before, but there are other mechanisms by which change to DNA occurs throughout generations. So we're going into population because evolution occurs at the population level. So that's, at least that's what we're looking at. We're looking at the genes in the population that make that a different looking population than any other. So leopards are a different looking population than lions, although they're all cats, but they're different enough. So these are Cuban tree snails. This is one population. And I love this picture because it shows the variety of morphology, shell morphology of this population. So along comes some kind of selective pressure, like a predator could be a bird. And this bird might pick off, you know, the ones it can see very easily. Maybe it's the white ones. And there's a gene for white shell color. Well, if the bird picks off all of those white shell color snails, that white allele may indeed disappear from the population. So, whoopsie, sorry. This is an example of grasses. And in this area here, this has mine tailings. So those are minerals and excess kind of things that come from mines that wash out of the mine area. And these grasses here were able to adapt to the toxins in the soil. So they thrive here. But these grasses over here, they don't have that. This population lacks the allele that is resistant to the toxins, essentially. So this is bent grass, it's called. Yeah. So they evolved in a relatively short space of time, too, into different populations. So population genetics, which is what we're talking about now, and we've already looked at population genetics up to now when we studied Mendel. So Mendel and inheritance, that's what population genetics is. So you really, when you study genetics, we study it at the two levels. This is a broader level, population genetics, and then we might look at it at a molecular level, molecular genetics, and we'll do that next term. So what it does, it combines the Darwinian's idea of selection and Mendelian inheritance. So population genetics is the study of genetic variation in a population. And the emphasis is on quantitative characters because that's what we can measure. Really, this comprehensive theory of evolution was synthesized in the 1940s using Darwin's work and Mendel's work and the work of other scientists as well. But until then, people didn't really accept that the theory of natural selection could drive evolution, such an enormous change. So the modern synthesis, as we call it, evolution, combines discoveries from paleontology, so those are all the fossils that we talked about, taxonomy, so that's Linnaeus. How do you spell his name? Linnaeus? Nope. That is not how you spell his name. Linnaeus. That might not be it either. Biogeography. So we looked at those two animals, the glider, an example would be the glider and the flying squirrel. two different continents, similar features, and that's called convergent evolution. Adaptations. Down, let's say, different lineages. So the flying squirrels came down a lineage of placental mammals, and the gliders came down a line of marsupial mammals. But the adaptations are similar, so it's called convergent evolution. So the importance of population genetics is that populations are the units of evolution. And that's because we're looking at a population being of the same species. And natural selection really is the most important mechanism of evolution and gradualism, gradual change. Of environment, a gradual change of mutations, leading to selection. I mean, let's get real here. If an animal or a plant or a fungus or any organism has a mutation, then it's probably not going to be good. And for the most part, mutations aren't beneficial, but sometimes they are. So that leads to, if it's adaptive, selection of the individual with that new mutation. We're going to talk about mutation in a little bit. So let's talk about allele frequency. Hang on, before I do that, there's one thing I wanted to say. That microevolution is the change in allele frequencies from one generation to the next. And it's not until there's an accumulation of all of these changes that it becomes macroevolution, where you could actually get speciation, a new species from an old one. So let's look at allele frequencies. Gene pools. Gene pools, a gene pool is all the genes, all alleles in a population. Do you remember what populations are? Have I explained that well enough?
Units of evolution?
Yes, units of evolution. And a population is an assemblage of organisms of the same species. So let's say we start off with 500 flowering plants in this population. Of those 500 flowering plants, 480 are red and 20 are white. Of the ones that are red, 320 are homozygous dominant, or big R, big R. 160 are heterozygous, big R, little r. And 20 are recessive, little r, little r. So there are 1,000 copies of the gene for color. We have 500 plants. All of those 500 plants have 2 alleles. So there are 100 copies of the gene, or sorry, 1,000 copies of the gene, or 1,000 alleles in the population. Because each of those 500 plants has 2 alleles for flower color. So here are the allele frequencies. So we're putting all the males and females together here. 320 times 2, because we've got 2 big R's there, 160 times 1, because we've only got one big R there. And that equals 800 dominant alleles, 800 red alleles, or 800 over 1,000. So our frequency is 0.8 or 80% of that particular allele. Let's look at the recessive allele. We've got 160 times 1. I better do that in a different color. Times 1 plus 20 times 2. And that gives us 200 little r alleles, or the white allele. 200 over 1,000 initial alleles gives us 20%. So that is our allele frequency for flower color in this population of 500 plants. Are you with me so far? All right. Excellent. Okay, so now I'm going to prove to you beyond a shadow of a doubt that without any natural selection, without any different anomalies like genetic drift, with completely random fertilization, we're going to have the same frequency of alleles in the next generation. So let's see how that works. So yeah, okay, so population, localized group of individuals of the same species. A species, a group of populations who have the ability to breed and produce fertile offspring. So all of these plants are able to reproduce and produce offspring. Usually the individuals near the center of a population are more closely related than members of other populations. I don't really know why that's in there right now. Did I miss one? I seem to be missing an entire slide. Well, that's just fine. Then, okay, ignore what's up here. Let's just do it. You can do that with me. So we're going to do a Punnett Square. The males and females are all mixed together with the same frequencies of alleles. So we're going to, we are going to breed, who are we breeding? We're breeding females that are heterozygous with males that are heterozygous. But we're just breeding in amongst the whole population of heterozygotes. And so the frequency of little r we said was 0.2. The frequency of the big R is 0.8 in the population. Now we're doing this at a population scale. Again, the big R is 0.8, little r is 0.2. Okay, so I didn't make these boxes big enough, but here we have, of course, our fertilization of an egg with a big R and a sperm with a big R, and we have a homozygous dominant individual. Here we have heterozygous individual, heterozygous, and homozygous recessive. We're just using the same principle with our Mendelian genetics monohybrid crosses, but we're using the whole population. And we know what the frequencies are of the alleles in that population. So here is our new population, our F1 population. So what have we got frequency-wise? Well, if we've got two big R's, what is the probability of getting two big R's there?
25%?
Not anymore, because we're looking at the whole population as opposed to an individual. So the whole population has 80% of those individuals have the big R there, and 80% have the big R there. So we're looking at the frequency of the population now. So that's what we have to multiply together. So then we've got 0.64.
I totally fell for it. Yep.
It was not purpose at all. So what have we got here for big R, little r?
.16.
Good-o. And here?
Same.
Same. And here?
0.4. Sorry, 0.004.
0.04.
0.04.
Right? Is that right, or is it just 0.4?
0.04.
Okay. Okay, good. Thank you. So, so how are we going to get the next generation? Well, we have a 0.64 big R. So 0.64 times 2, I think, I hope I do this right, I might not, plus 0.2 times 1 plus, no, that's wrong. See, I'm doing it wrong already. Rats.
We got to only consider the capital R's now, right?
Yeah.
Okay.
Yeah, only the capital R's. So,
This is 0.64 times 2, and then plus 0.16 and 0.16 again, I believe.
I think so. Okay.
And that is...
Can someone work that out? And then times 1. Huh.
1.6. But there's something wrong, isn't there?
There's something a little wrong. So we're looking at the genotype and allele frequencies. I don't know if I can work this out off the top of my head. I should be able to, but I don't know if I can. Yeah. How does that work now? So what is the bottom line then? 0.16, 0.32 plus, so that'd be 0.40, right?
That is...
So if we were to multiply this by 100, we would get 160. If we were to multiply this by 100, we would get 40. And if we divide that by 2 and divide this by 2, we've got 80 and 20. So by some roundabout way, mathematically, I can't fathom at this particular moment in time. And I wish I hadn't recorded this lecture, but we did end up with 80% of our big R's and 20% of our little R's. So that was the same frequency as we had before. So
I think it's
somehow
because we don't have the number of the population. No? Then we have to start with that number, and then once
Not
we
really.
have... No, we don't?
Not really, no. No, for some reason, mathematically, I mean, it did work out in the end. We just had to put in a couple of extra steps. I'm not
What was
entirely
the
sure why, but I think it's
500?
because we're looking at percentages as opposed to right proportions. So that's all it was. We just had to convert to proportions. But why we divided by 2, I'm not exactly sure about that.
The 500 that we had before that we started with, that was multiplied by 2. So I'm not sure.
Ah,
Are you going to lose all this, all the notes if you go back to the previous slide?
possibly.
Oh, actually, it should stay. This one, and then it divided 800, which is the total number of capital R's, by 1,000, because we started with 500 flowering plants, and because they are dividing, then we make it 1,000.
I'm not sure. This doesn't bode that well for what we're going to do next. But it's quite funny. I'm just glad I'm just glad that somehow it worked out. Okay, how about this? Let's just accept that the allele frequency in the next population will be the same if everyone interbreeds the way, just completely randomly. I'll just
Illegal
say
math.
that for now. Yeah, we'll leave out the math. I'm about to throw at you a bunch of math. It doesn't seem fair. I might not upload this lecture.